Hi dear students, I welcome you all for this YouTube platform of Sree Balaji Medical College and Hospitals. I am Dr. Shanbag Lalita, Associate Professor of Biochemistry. Today in this session I am going to teach you about cell membrane and fluidity. At the end of this session I am sure you will be able to enumerate the types and functions of the cell membrane, its distribution and its chemical composition and its properties of the cell membrane. So before going into detail, let me give you a brief introduction about cell membrane. So cell membrane is going to give the cell shape and it's going to limit the size of the cell. It also maintains the inner environment of the cell. It regulates the transport across the cell. It also allows the interaction between the cells. It also interacts with the external environment and brings changes accordingly inside the cell. It regulates the cell transport. It also has receptors to bind hormones, and it also has enzymes which are activated when receptors bind with hormones. And please keep in mind the cell membrane is always selectively permeable and it is not a rigid nature. It is very fluidic in nature. So there are different types of cell membrane present in your body. Usually in mammals we call it as plasma membrane. The membrane which is surrounding the nucleus is known as nuclear membrane. The membrane which is surrounding the mitochondria is known as mitochondrial membrane, which is again categorized into outer and inner mitochondrial membrane. There are special membrane present in endoplasmic reticulum, and the membrane present in the nerve cells are known as myelin. So coming to its chemical composition, plasma membrane consists of three biomolecules: lipids, proteins, the third one is carbohydrates. The composition may vary from one type of the membrane to another, but usually in plasma membrane 40 to 45% is made up of lipids, 50 to 60% is made up of proteins, and carbohydrates is present in a very minimal amount, that is nearly 1 to 10%. So if you see in this tabular column, usually I told plasma membrane 43% will be lipids, 49 to 50% will be proteins, and carbohydrates are present in a very minimal amount. If you take nuclear membrane, if you see the distribution, protein component will be more in nuclear membrane than carbohydrates and lipids. If you see mitochondrial membrane, there are only trace amount of carbohydrates present in the mitochondrial membrane. If you see myelin, then lipid component will be more in the myelin cells compared to proteins and carbohydrates. So coming to the first biomolecule, which is lipids. This lipids is going to give a basic structural component of plasma membrane. It forms a lipid bilayer. There are four major classes of lipids present in the cell membrane. The most abundant one is phospholipid. The next comes a sphingolipid, and then glycolipid, and another form of lipid which is known as cholesterol. So all of them are amphipathic molecules which is going to possess both hydrophobic and hydrophilic properties. So if you see here in the hydrophilic head end you have glycerol and phosphate. in hydrophobic tail end consists of saturated fatty acids and unsaturated fatty acids. Saturated fatty acids is nothing but a fatty acid without a double bond. The unsaturated fatty acids is nothing but a fatty acid with a double bond. So both these form the phospholipid bilayer of the cell. So usually the hydrophobic tail ends are stacked in the interior of the lipid bilayer, while hydrophilic head groups will be face the exterior of the lipid bilayer. So phospholipids are the most abundant lipids, as I told you in the previous slides. Based on the types of the alcohol moiety present in the phospholipids, it is classified into glycerophospholipids and sphingolipids. So glycerophospholipids is again categorized. The examples of glycerophospholipids are phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, phosphatidylinositol. Sphingolipids, the examples are sphingomyelins, cerebrosides and gangliosides. So the plasma membrane is usually asymmetric in nature, that is, it contains different types of phospholipids in the outer and inner leaflet, which is going to increase the fluidity of the cell membrane. Usually in the outer leaflet, you have phosphatidylcholine and phosphatidylethanolamine. In the inner leaflet, you have phosphatidylserine, phosphatidylinositol and sphingomyelin. So the coming to the next major lipid in the lipid bilayer is cholesterol, which is found dispersed between the hydrophobic tails of the membrane phospholipids. This cholesterol is also having a hydrophilic head end and hydrophobic tail part. The hydrophobic tail part is stacked inside the hydrophobic tails of the phospholipid layer, and the hydrophilic end will be dispersed between the hydrophilic head groups of the phospholipid bilayer. So the cholesterol acts as a fluidity buffer. It regulates the fluidity of the plasma membrane. So when the cell membrane is exposed at higher temperature, then this cholesterol is going to inhibit the movement of phospholipid fatty acid and reduces the membrane fluidity. When it is exposed to cold temperature, then cholesterol interferes with fatty acid interactions, acting as antifreeze, and it maintains the fluidity of the cell membrane. So coming to the next biomolecule, which is proteins. So according to their position in the plasma membrane, the proteins fall into three main categories. The first one is integral or intrinsic proteins, that is, it is deeply embedded inside the cell membrane. Peripheral or extrinsic proteins, these are present one side or other side of the membrane and it is very loosely attached. And the next one is the transmembrane proteins, which is going to span the whole breadth of the cell membrane. These transmembranes usually serve as receptors for hormones, neurotransmitters, ion channels, growth factors, etc. So apart from these integral, peripheral and transmembrane proteins, certain membranes like RBC cells have special types of proteins which is present in the integral part and also in the peripheral region. So the special integral protein present in the RBC membrane is glycoporin and band 3 protein. So this glycoporin acts as a blood group antigens and it also acts as a site for influenza virus attached to RBCs. In the band 3 proteins, in the lung capillaries, exchange of bicarbonate ions for chloride ions occurs through this protein. So special peripheral proteins present in RBC membranes are spectrin, actin and band 4-1 proteins, which is attached to ankyrin, a part of cytoskeleton, a peripheral protein which is going to form the skeleton of the RBCs and it is going to stabilize the RBCs. So clinical significance associated with this special proteins or hereditary spherocytosis and hereditary elliptocytosis. Both are a genetic condition. So it occurs due to mutation in the genes encoding for protein spectrin or absence of band 41 protein, which is going to result in abnormal shape of RBCs. So the patient will have increased hemolysis, anemia and jaundice. So coming to the next biomolecule, carbohydrates. These carbohydrates are short unbranched or branched chain of sugars. It will be either attached to proteins. If it is attached to protein, then it is known as glycoprotein, or it attached to the polar ends of the phospholipids at the external surface of plasma membrane, which forms glycolipids. This carbohydrates usually serves as cell identification tags. So it helps in cell-cell recognition, cell-cell signaling and cell-cell interaction. Apart from these lipids, proteins and carbohydrates, there are also special molecules present in the cell membrane, which is known as lipid rafts and caveoli. Lipid rafts are enriched with cholesterol and sphingolipids, which is present in the outer leaflet of lipid bilayer. The main function of lipid rafts is involved in cell signaling. Caveoli, it is derived from the lipid rafts. It is a special protein, helps in the formation of lipid rafts. Function, it is mainly involved in cell signaling. So how the lipid bilayer is formed? Not all the lipids will form a lipid bilayer. Only glycerosphingolipids and sphingolipids will form a lipid bilayer, because whenever the hydrophobic tails end and the hydrophobic heads are equal, then it forms a lipid bilayer. So glycerosphingolipids and sphingolipids fulfills this category, so it spontaneously forms a lipid bilayer whenever it interacts. And also the hydrophobic effect and the solvent entropy provide the driving force for the lipid bilayer formation. So the fluidic nature of plasma membrane is proposed by Singer and Nicholson in 1972. This is the widely accepted model, which is known as a fluid mosaic model. The fluidity is due to the individual phospholipids and some proteins can move sideways, that is laterally, in each layer. Therefore, it is fluidic in nature. The mosaic appearance is due to the range of different proteins resting on the surface through the phospholipid layer gives it a mosaic appearance. So if you see the plasma membrane, when it is in low temperature, then it is like gel-like organization will be seen. The polar heads are tightly packed, the tails will be regular and membrane will be thicker. So when it is exposed to higher temperature, then the lipid bilayer will be fluid-like organization, and the polar heads are loosely packed and the tails will be distorted and membrane will be thinner. So this is the lateral movement of the plasma membrane. Usually you have lateral movement. Sometimes you will see a flip-flop type of movement also, which is a very, very rare condition. So why the fluidity is more important? The membrane must be fluid to work properly. Solidification may result in permeability changes and enzyme deactivation. The unsaturated hydrocarbon tails enhance membrane fluidity because the kinks at the carbon-to-carbon double bonds hinder the close packing of phospholipids. Thereby, it maintains the fluidity. To summarize it, cell membrane is going to give a shape to the cell, and it also limits the size of the cell. It is selectively permeable and fluidic in nature. It also interacts with the external environment and brings changes accordingly inside the cell. It also helps to maintain the inner environment of the cell. It also regulates the cell transport across the cells, and it also interacts with the other cells between the cells. And it has three major biomolecules: lipids, proteins, carbohydrates. The major abundant lipid found is phospholipid, which forms a bilayer. Proteins are categorized into extrinsic, intrinsic, and transmembrane proteins. Carbohydrates, which is distributed minimally, is going to act as cell identification tag. To remember cell membrane in a simpler way, you can think of your house. The wall of your house is the lipid bilayer. The main entrance door is the transmembrane protein. The windows are peripheral proteins. The small door inside your house is the intrinsic protein. The house ID or your house name or house number is your carbohydrates. Thank you all for the patient listening.